Hello, everyone. It's a pleasure to be here today with you, even virtually, to share with you our work on developing a simple, fast, and inexpensive test for the viruses that causes COVID-19. 8 months after the pandemic hit the US, 1 out of 350 people in New York City have died from COVID. That's 1 out of 350. In the US as a whole, things are not much better, with over 250,000 deaths so far and close to 1,000 deaths per day since July. Now, one of the main reasons the pandemic has been so out of control in much of the Western world has been the lack of appropriate testing to identify not only who is sick with COVID, but who is contagious. And when deaths began to grow exponentially in New York in April, and it was clear that inadequate testing was a big part of the problem, a group of us, faculty, postdocs, students, and staff here at the Zuckerman Institute and throughout Columbia got together to develop a simple, fast, and inexpensive test for the coronavirus. While none of us were experts in diagnostics or virology, we thought that our combined expertise in molecular biology and chemistry would result in a synergistic team. So first, let me tell you why testing is so important for preventing the spread of the virus. Now, 4 out of 10 people who are infected with the coronavirus do not have symptoms. And these people without symptoms actually contribute to 50 to 60% of onward infections. Therefore, isolation of people with symptoms does not stop the majority of contagions. So here's the typical progression of an infection with the coronavirus. After exposure to the virus and infection, there is an incubation period that typically lasts 4 to 5 days. And this period is then followed by symptoms that vary in length and in severity from a handful of days to 10, and even the long haulers, which are now called to have symptoms lasting for several months. So if we now plot how much virus there is in an infected person's upper respiratory tract, the nose, the mouth, and the throat, we see that it increases rapidly and peaks a day before symptoms develop. And this high viral load preceding symptom onset is a reason why a large fraction of contagions occur before the symptoms develop. While the exact relationship between viral load and infectivity is not yet known, there does seem to be a strong correlation between the two, and data indicating that people with low levels of the virus are not really very infectious. In line with this, people with mild to moderate symptoms are almost never contagious 10 days after symptoms start, because their viral load has already decreased substantially. So once you know this, it becomes apparent that testing can have multiple purposes. A medical goal to know the best course of treatment and prognosis for someone with symptoms, in which you want to make sure you have a very sensitive test. You can also have an epidemiological data goal to know how many people are infected through time, and a third goal to detect infectious people and prevent them from spreading the disease, regardless of the presence of symptoms. For the medical and epidemiological purposes, you want a high sensitive method so you don't miss any cases. But to detect infectious people, you can use less sensitive tests that detect people with the high viral loads associated with infectivity. Now, the PCR tests are the most sensitive methods and detect most cases. But the drawback is that many people continue to test positive long after they're no longer infectious. And this limits its utility as a method to minimize contagions without asking too many people to isolate when they are still positive by the PCR test, but their levels of the virus are so low that they will not infect anyone. In fact, currently half of the PCR tests that return positive are returned to the user. The viral load is actually so low that the person is unlikely to be infectious. In contrast, there are other types of tests which are often much faster to perform and inexpensive compared to the PCR test. And however, they're less sensitive, but the positivity in this test, as you can see here, overlaps and correlates better with the infectious period. So it is not only important to select the most appropriate test, but also that it is performed at regular intervals, since there is no guarantee that if you tested negative on a Friday, you will still be negative on a Monday, for example. So what is really the effect of testing frequency and of test sensitivity on decreasing the spread of the virus in a population? So here are the results of models published a couple months ago that compare different testing strategies in their effectiveness of preventing people from infecting others by asking them to isolate if they are positive. Because many people are contagious before they develop symptoms, and also not everybody isolates as soon as symptoms develop, without any testing, only 15% of infectiousness is removed from a population if you rely only in self-isolation after symptoms start. In contrast, daily testing has the largest effect on eliminating infectiousness, identifying essentially everyone, regardless of whether the test is the more sensitive PCR test or a less sensitive rapid test. Testing 3 days a week misses around 10% of infectious people, and testing 14 days is not very effective. Importantly, the cost benefit of these testing strategies really depend on how many people in the population are infectious and in the effectiveness and adoption of other measures to minimize spread, such as wearing masks and hopefully soon also vaccination. So here we see that in addition to the testing frequency, whether it happens daily or 3 days a week or weekly or every 14 days, the lag in the return of the test after the test is performed is crucial. It's also very important. We see that a lag of even 1 day or 2 days affects the effectiveness or the testing on isolating infectious individuals. Ideally, you want to return the results on the same day that the test is performed. So people, if they have a positive test, will stay home and stop spreading the disease in the other days that they're waiting to hear from a result. So what if testing could become part of your daily routine, or at least make it testing so easy and widely available that people can get tested daily at school or at work? So this is what we set out to do, to develop a simple and inexpensive test that didn't require fancy laboratory equipment or technical expertise, and that relied on materials that were in short supply, for example, how that happened early in the pandemic. So our goal was to develop a test that was fast, painless, simple, inexpensive, and accurate in detecting infectious people. First, to understand how testing works, it is helpful to know a bit more about the virus itself. And here is a beautiful illustration of the SARS coronavirus 2 that causes COVID-19. The virus has a very simple and elegant structure with proteins on the outside that it uses to attach to your cells and infect you. And its genome, shown here in red, inside a membrane. And this genome is covered in a protein here shown in blue. So there are 2 main ways to test for the presence of the virus in a person. One way is for looking at viral proteins as part of these rapid antigen tests. And these tests are usually fast, but it is still unclear if they are sensitive enough to detect most infectious people. The other way is to look for the viral genes, which in the case of the coronavirus are made up of RNA. And one such test, the most widely performed test, is the PCR test, which makes many millions of copies of the viral genome. And this amplification of the viral genome makes PCR tests very sensitive. But on the other hand, they're also too expensive and slow for daily testing. And one of the reasons they're too complicated for testing outside of a laboratory setting is that they require expensive equipment necessary for quickly cycling temperatures. As you see here, it requires 25 to 50, and then 45 times going to 95, 55, 95, 55, 45 times to amplify this DNA. And in addition, it needs expensive equipment to detect the fluorescence that is used to detect these amplified DNA molecules. Now, an alternative to the PCR test is the use of LAMP technology. And this is a fantastic feat of molecular ingenuity, which also amplifies viral genes, but much faster and at a constant temperature, obviating the need for expensive equipment. So this video shows how LAMP technology works. So here it starts with the RNA of the virus that is converted by DNA. And then the whole reaction happens at constant temperature while you provide some of these reagents. It all happens in the single tube at a single temperature. And within minutes, the DNA starts to become amplified through this reaction. there's this formation of these loops, and then amplifying in the backward direction until these dumbbell structures are formed, these lamp dumbbell structures, which are then able to bind to each other and to bind to more of these primers. And this leads to the formation of these concatenators and these products that amplify exponentially, all again continuously at a single temperature. And sorry. All right, so 5 years ago, Nathan Tanner at New England Biolabs developed a system to be able to detect whether there was positive amplification of, for example, viral DNA or anything you're trying to amplify. If there is amplification, this reaction is yellow, and if the test is negative, if there's no amplification, it's red, giving a very easy readout, which contrasts with the PCR, in which, as I told you, this real-time PCR often requires this fluorescence readout. So LAMP technology was adapted for the detection of the coronavirus early in the pandemic, but still existing methods rely on laboratory instruments, including pipettes and centrifuges. And what we set out to do then was to further adapt this technology so it wouldn't require these instruments and so that it could become as simple as possible. And because saliva is easy to obtain painlessly, and because other scientists had shown that it contains as much, if not more, of the virus than the swabs that are typically used in your nose or in your throat, we decided to use saliva for our test. However, saliva is a complicated fluid because it can, in addition to the virus, it can have remnants of food and digestive enzymes and your own cells, bacteria, and other viruses, in addition to the one we're trying to find, many electrolytes and pH buffers, along as mucus and things that make it viscous and difficult to work with. So laboratory tests that for the coronavirus that use saliva first purify the RNA, and that is the process that really requires the centrifuges and the other lab equipment to isolate and purify the RNA from the virus. So what we did was to set out to find ways to enrich for this RNA and to chemically inhibit the other components of the saliva without any lab equipment. And after many months of trial and error, we finally succeeded, and our test now looks essentially like this. You take some saliva into a tube, you add some chemicals, you heat it up, you remove those chemicals and then add new ones, and then you heat it up again and for further 45 minutes until you see if the reaction turned pink or yellow. And we can do two things. One can heat it up in a pot of hot water, but we have also developed this apparatus that with a single press of a button, it's a simple and inexpensive apparatus, a simple press of a button, it'll heat it up to the required temperature. So to validate our test, we took saliva from people with varying levels of the virus, shown here on the x-axis. Lower CT values indicate a higher viral load, and these higher CT values indicate that these people had higher viral loads as measured by the standard PCR test. And you can see is that our LAMP test, here we can see we can detect 100% of the people who have a CT cycle of 20 or lower, or 24 or lower. And then we detect most people, 90% of people who have 28 or lower, and then the performance tends to decrease with the higher viral loads. And this is very exciting because, as I told you earlier in the presentation, low viral titers usually means no infectiousness. And these are the results of a study from other people in which they were correlating these CT values, the amount of viral load in a person's nose and saliva, with the ability to culture the virus in the laboratory. And you can see when people have high viral titers, you get most of those correlate with having a lot of positive cultures, and low viral titers means very few of these samples actually carry the live virus. So we were very happy to see that our two tests were our test and these results were very well aligned. So our assay is therefore now very robust, and we are currently working on simplifying even further to make it as useful and widely applicable as possible. And with that, I want to thank the large group of volunteers who helped in many different aspects of this work, from writing code and developing designing reagents and actually testing people, and especially the people highlighted in bold and with their large names, and particularly David Ing, who made especially big contributions to the work I told you about today. Thank you very much.